of Breathing, brought to you by Gabby Nunez and Courtney Rosales. In order to function, organisms are divided in several body systems. Because the systems are not isolated, it is important to study and understand all systems individually and as a whole. This laboratory focuses on the interlinked functions of the respiratory and circulatory systems that maintain a neutral pH in the blood. The study and research of this process is necessary in order to understand a key mechanism for maintaining homeostasis. The circulatory system is responsible for the transportation of substances between all the other systems in the body. The circulatory system is divided into two parts, systemic circulation and pulmonary circulation. The primary function of the respiratory system is to supply the blood with the oxygen so that it is transported to all parts of the body through the circulatory system. The respiratory system does this through breathing. When we breathe, we inhale oxygen and exhale carbon dioxide. The inhaled oxygen passes into the alveoli and then diffuses through the capillaries into the arterial blood. Meanwhile, the waste-rich blood from the veins releases its carbon dioxide into the alveoli and is exhaled out of the lungs. The carbonic acid bicarbonate buffer is the major buffer used by the body to regulate pH of extracellular fluid. The carbonic acid bicarbonate buffer is constantly adjusting the blood's pH of 7.4 to a pH level of 7. When an addition of protons to the blood due to strenuous exercise is added, it becomes too great for the buffer to effectively control pH levels alone. Hence, the organs in the circulatory and respiratory system step in by controlling the amounts of carbon dioxide and HCO3. The following slides will give all the details pertaining to the execution, results, and discussion of this laboratory. The major objective of this lab was to understand the acid-base balance in the body and examine lung capacity. Before starting the laboratory, it was hypothesized that the body's breath contains a higher level of carbon dioxide than the air in a room. Additionally, it was hypothesized that the body exhales more carbon dioxide after exercise than while at rest. To test these hypotheses, 3 experiments were carried out. Based on what was hypothesized, it was predicted that in the first experiment, the beaker testing room air would take longer to turn cloudy than the beaker testing human breath, and that in the second experiment, the beaker testing human breath after exercise would take more ammonia drops than the other 2 beakers.
This experiment was completed according to the procedure outlined in the BB2903 Lab 5 from Buchholz and Hunter 2011. There was one procedural change in Experiment 2 for the effect of exercise on carbon dioxide production within the body. Instead of adding 10 drops of phenolphthalein to the beaker, phenolphthalein was already added to the source of water, and 200 milliliters of phenolphthalein was used to complete the experiment. The objective of the first respiration experiment was to compare the amount of carbon dioxide in room air versus exhaled air. It was hypothesized that exhaled air would contain a higher carbon dioxide concentration than room air. To test the hypothesis, room air and exhaled air were bubbled into lime water, which is concentrated calcium hydroxide that turns cloudy when it comes in contact with carbon dioxide. The room air was bubbled with a pipette and a pipette pump in 50 milliliters of lime water for 1 minute. After 1 minute, the water appeared to be only slightly cloudy, indicating that the carbon dioxide content was extremely low. The lime water before bubbling and after bubbling room air is seen here. The lime water only appears to be slightly cloudy. Exhaled air was also bubbled into 50 milliliters of lime water for a minute through a straw at a constant or similar rate as that of the pipette pump. After only seconds of bubbling with respired air, the lime water turned extremely cloudy compared to the room air sample. The lime water experiment showed that the water with bubbled respired air turned cloudy faster than the lime water bubbled with room air. This concludes that the respired air contains more carbon dioxide than room air. Here you can see the limestone sample before respiration and after respiration. It is very clear that the amount of carbon dioxide in respired air is higher because of how cloudy it is. Shown here is a table to record the results and observations from this experiment. After 1 minute, the room air sample was slightly cloudy, and after 1 minute, the respired air sample was extremely cloudy.
The second experiment aimed to examine the amount of carbon dioxide produced at rest and after exercise, using phenolphthalein as an indicator of carbon dioxide level. Phenolphthalein changes from pink to clear under acidic conditions, as when carbon dioxide is bubbled through and produces carbonic acid. During the experiment, 3 beakers were filled with 200 milliliters of phenolphthalein and water solution. The first beaker tested the amount of carbon dioxide in the room air. This was done by bubbling air into the solution with a pipette pump until until the solution became clear. The slide presents the beaker with the pink liquid before the pump in air and after 94 seconds when the liquid turned clear. The second beaker tested the amount of carbon dioxide exhaled at rest. Instead of pumping with a pipette, air was exhaled through a straw into the solution. The slide presents the beaker with the pink liquid before the air was blown in and after 23 seconds when the liquid turned clear. The third beaker tested the amount of carbon dioxide exhaled after strenuous activity. For this one, air was exhaled into the solution after 2 minutes of exercise. The slide presents the beaker with the pink liquid before the air was blown in and after 17 seconds when the liquid turned clear. The amount of carbon dioxide in each beaker was then estimated by adding drops of ammonia, which decreased the acidity of the solution, to the beaker until the solution changed to a light pink color. The number of drops was then recorded. After the drops of ammonia were added, the amount for each beaker was normalized using the equation displayed in this slide. Table 1 shows the results of this experiment. As observed in this table, the beaker with exhaled air after exercise took the shortest time to clear and had more drops of ammonia added. Since the presence of carbon dioxide turns phenolphthalein clear, the fact that it took the shortest time to turn the beaker with the exhaled air after exercise clear proves that this beaker had more carbon dioxide introduced to it. Likewise, the fact that this beaker also needed a greater amount of ammonia drops proves that there is more carbon dioxide present in exhaled air after exercise than in exhaled air at rest and room air. However, in order to be more certain that the levels of carbon dioxide were truly different, an analysis of variance ANOVA was performed. The ANOVA was performed on the normalized number of drops data for the Wednesday evening section in order to see if the difference observed between the levels of carbon dioxide under different conditions was real. The ANOVA returned a p-value of 5.74 times 10 to the negative 5. This means that the difference in the data was statistically significant. Therefore, the results shown above did follow the pattern predicted, proving that exhaled air has more carbon dioxide than room air and that the body exhales more carbon dioxide after exercise than at rest. A possible explanation for the higher level of carbon dioxide in breath than in room air is that during the breathing process, inhaled oxygen is exchanged for the carbon dioxide produced by cellular processes. As explained previously, this carbon dioxide in the blood produces an increase in blood acidity. In order to neutralize the pH level of blood, the brain signals the respiratory organs to take the carbon dioxide from the blood in the alveoli and exhale it. As for the difference between the exhaled air before and after exercise, it can be assumed it exists because the body needs more oxygen to perform the extra activity, and the increase in oxygen is compensated with an increase in carbon dioxide exhaled.
The third respiration experiment that was performed in lab was a vital capacity experiment. Vital capacity is the amount of exchangeable air in the lungs. The vital capacity is the sum of the inspiratory reserve volume, which is the total amount of air the average person can forcibly inhale, plus the expiratory reserve volume, which is the amount of air that the average person can forcibly exhale, plus the tidal volume, which is the total air that moves in and out of the lungs during breathing. The vital capacity can be measured by using a spirometer, which records the amount and rate of air exhaled or inhaled. For this experiment, each partner forcefully exhaled 3 times into a cardboard tube at the end of the spirometer. During exhalation, each partner covered their nose to ensure that the air was only coming out of the mouth into the tube. The gauge would then show the partner's vital capacity. The following is a table displaying the results from the vital capacity experiment. The table shows the results from each trial, the average, the normal vital capacity for someone of that height and weight, and the standard comparison, or how close that person is to the normal vital capacity. Courtney is extremely close to the normal vital capacity of someone her weight and size. Courtney's vital capacity is 98.98% of what it should be, whereas Gabby's is 76.67% of what it should be, according to her weight and height. Figure 19 shown in the slide is a visual representation of the normal distribution of vital capacity of the BB2903 class. The mean of the class vital capacity data was 3.46 with a standard deviation of 0.831. Courtney's vital capacity fell within 1 standard deviation of the class average, further proving that her vital capacity is normal for her size and weight. Gabby's vital capacity also fell within 1 standard deviation. After analyzing the results of these experiments, it was concluded that due to the exchange of gases that occurs inside the body with the joint effort of the circulatory and respiratory system, there is a higher level of carbon dioxide in the air exhaled than in a typical room air. Furthermore, it was also concluded that the body produces more carbon dioxide after strenuous activity due to the increased need for oxygen in the blood circulation and its subsequent increase in carbon dioxide exhalation. This information is relevant because it deepens one's understanding of the way the organism works. It helps to clarify the interactions between organ systems.